Were they?
I mean, if she got 6 apples for $4,
Hey man, well we can.
why do
Yeah, I didn't put a tab in the lab for the price of the apples, but we could calculate. We're going to measure the mass of them. And we could calculate the price per gram of these apples. See which apples are commanding a premium on the open market. That would be interesting to me. And then we can do water weight and see whether you're just buying water or whether you're actually buying like dry mass. The economics of apples. Is that okay? The fan? Can you guys hear me? All right. Okay, so from up until this point, I've been doing a lot of history, and I'm going to begin to switch gears slowly, not that slowly, into this is a biochemistry class, and I'm going to begin to give you more actual biochemistry and a little bit more actual meat to the science. As we do that, I'm going to try to build your foundation. I realize that none of you are science majors, so I'll try to give you all the tools along the way. For those of you who've had much of this before and for which it's review, great, I'm reviewing for you. And for those of you who haven't, stop me when you need to stop me to make sure that you're absorbing what I say. I am very conscious of the fact that I don't have a textbook for you. There is no textbook that covers the breadth of the material that I want in the not super deep depth that I'm going. So this material, you're going to have me and the lectures as your resource plus whatever. If you're not understanding a concept, you reach out to me or you look to the internet. There's all of this stuff is very well described on the internet. All right, so photosynthesis. We were talking about carbon, and we're going to be leaving carbon and coming back to it at different points. But the story of carbon, I'm going to take carbon through the plant fixation of carbon, photosynthesis. All right? So the formula for photosynthesis, it was the last thing I think I talked about last time, is that equation up there at the top. It's essentially where carbon dioxide gas is pulled out of the atmosphere and along with water that's absorbed from the root system of whatever plant, and using the energy of sunlight, creating carbon-carbon and carbon-hydrogen bonds, both of which are high energy bonds. I showed you that table last time that said that carbon-carbon bonds were about 89 calories or kilocalories per mole, and that carbon-hydrogen bonds were approximately 100 big C calories per mole, thus releasing oxygen, and the carbon that's been fixed takes the form of carbohydrates. All right, so these carbohydrates are the storage molecule that gets that carbon-carbon bond and carbon-hydrogen bond energy transferred into a molecule called ATP, which we will learn more about in a little bit. Here are the chemical structures of glucose, which is the simplest hexose. Aldohexose is what it's called. It's a carbohydrate. It's a monosaccharide. Mono meaning one, saccharide meaning sugar. It's a single sugar ring, glucose, and it's the energy, the carbon-based energy chemical that is the primary currency of energy in the body. It's circulating around in your blood. It can pass the blood. It's one of the few molecules that passes the blood-brain barrier into your brain. Your brain needs it for fuel. Glucose itself is just a building block for more complex carbohydrates, larger polymers. Polymer meaning many units. So these polymers can be amylose. Polymers of glucose are amylose, which you know as starch, vegetable starch, and like a potato, for example. And here you have, on the bottom, you see the chemical structure of that. And that's essentially repeating units of glucose that are hooked up via what's called a glycosidic linkage. We'll learn more about that later. I'm going to give you some basic chemistry first so that we can, when we get to the animal side, the human side of glucose, you can understand that. Glucose is attached in this chain by something called an alpha-1,4 linkage. Alpha, if you look at that, I need to get myself a pointer, but if you look at the oxygen that's sticking down, that's part of that bridge off the front of the carbohydrate to the left in amylose, that's axial. That means it's what's called an alpha. It's an alpha hydroxyl group. Cellulose, on the other hand, is identical to starch, but it's a beta linkage, and you can see that that oxygen is pointing up in that chain right there in cellulose. So cellulose is a beta-1,4, glucose to glucose, and amylose is an alpha-1,4, glucose to glucose. Cellulose, we can use starch as an energy source because we have the enzymes to hydrolyze or break that alpha-1,4 linkage, but we do not have the enzymes to break the beta-1,4 linkage. In fact, most animals don't. I was a carbohydrate chemist. That's what my graduate degree is in. And I studied these molecules. I used to know a guy who worked at Cornell, who was a theoretical scientist. This is a little bit of a story, a side story. And he, before he came to Cornell, was working at Los Alamos, down in the beehive of scientists that the government has down there. Los Alamos is one of the principal research facilities that our U.S. government maintains. And you may wonder why a carbohydrate chemist is working for the government. I mean, Los Alamos is where, I don't want to breed suspicion of the government, but that's where a lot of the awesome, spooky science that the government does happens, I guess. So anyways, what he was doing was trying to engineer a bacteria that rapidly hydrolyzed that beta-1,4 linkage, freeing the glucose in cellulose. Like when you eat, has anyone chewed on a piece of wood, stuck wood in your mouth? It doesn't really taste sweet, does it? It doesn't at all. But if you were to stick some starch in your mouth and let it sit there for a little while, it would get sweeter and sweeter with time, because you have amylase, which is the enzyme that our body makes that'll catalyze the breakdown of that bond. The project he was working on was developing a bacteria that can turn wood pulp into glucose that would be available for fermentation, that could be used to make ethanol as a fuel additive. Sounds like a good idea, right? There's a lot of wood pulp out there. Instead of having to grow corn or whatever to get the starch, there's a lot of things that grow cellulose. But if you think about it, what if that bacteria got out into the biosphere? What would happen? What if it got out of the petri dish? Imagine a bacteria that just is ripping through cellulose rapidly out in the biosphere. Our trees would suddenly be melting into a puddle of glucose. All right, so just a cautionary tale. He eventually gave up on the project and left Los Alamos. But cautionary tale about science and the need for the public to understand it and help guide the ethics of different scientific research. All right, so photosynthesis. Anyways, it's light. The energy from light is captured and used to reduce carbon dioxide to sugars. All right, and how does that happen? It happens principally via a molecule called chlorophyll. We can see a molecule of chlorophyll on the left-hand side there on top of that leaf. And it's got this long, greasy tail. The ME is not referred to you as in me, but it refers to methyl groups, kind of like a methane group that's attached. So very greasy, hydrophobic tail. And we'll learn what that even means in a little while when we talk about proteins. And then this big ring, reminiscent of a porphyrin ring that has magnesium coordinated in the middle of it. And this is the unit that catalyzes the formation of carbon-carbon bonds and the fixation of carbon dioxide. This porphyrin ring with the magnesium, this chlorophyll, absorbs light across the visible light spectrum. However, it mostly reflects light in the green wavelengths. So if you look at that mountain range dotted line there on the left-hand side, you see how there's a valley right around green. That's the light that it's not absorbing. That's the light it's reflecting. That's why leaves look green to us. It's reflecting off the green light. And all the other wavelengths, it's absorbing. Yes, please. Loud and proud. That thing is loud in my ear. Thank you. Perfect transition to the right side of the graph. There's more than one chromophore. A chromophore is a molecule that's going to absorb light energy. There's more than one chromophore that is in a leaf, in a plant. And some of them have totally different absorbance profiles. So, for example, look at this phycoerythrin in the middle there, that red peak that is absorbing all the way through the green light and blue light part of the spectrum. But it has a valley, it dips out where it's red. That chromophore comes on late in the season and appears red to us. You can see others, such as chlorophyll B, which are going to make leaves look more yellow as the peak is high in the red to catch the red light at the end of the season, but it's very low in the yellow part of the spectrum. All right? Does that make sense to you guys? So there's all those different chromophores can be found in the plant kingdom. For example, the carotenoids, you can see that word there. It has caret in the beginning of it. That chromophore looks orange. It looks orange because it absorbs in the blue and the green part of the spectrum, but it does not in the orange part of the spectrum, etc., etc. Phycocyanin. That's a bluish pigment. Sometimes you'll see bluish pigments in plants, not often. But, for example, in a blueberry, for example, this is something that absorbs color in yellow, orange, and red, and not much in the blue and the purple. Okay? This is what gives plants their color. You guys following me here? Everyone with this? Yeah, Christian.
So would like a Japanese maple which has something darker that represents the
Sure, I don't actually know the specific chromophores in a Japanese maple, but I could draw what I would guess would be their profile. It's going to probably have things on the blue end and the red end would be some troughs. All right, photosynthesis. This is about as complicated as it's going to get. Yeah, Ann. Yeah, you're going to have to speak really loud because I seriously can't hear over that fan. Yeah. They're going to have slightly different carotenoid profiles. So the carotenoids are a family. It's a large family of chromophores. And so there's going to be multiple ones, some that you're going to find in those red carrots, some that you're going to find in the yellow, some that may be in combination in the orange carrots. Does that make sense? Okay. Yeah, this is just a very simple, I mean, there are hundreds of chromophores, probably thousands. This is a simplistic diagram. All right, so photosynthesis. This is about as complicated as I'm going to get into photosynthesis. It gets really sophisticated, all the chemical steps. This is a very broad overview that I want you to understand because you're going to have to understand it for me to describe the difference between C3 and C4 plants. C3 plants would be cold-adapted plants, for example, a carrot or a cucumber would be C3, whereas C4 would be like corn would be a C4. And we'll talk about what that means, but first you got to understand what this is here. All right, the basics of photosynthesis. So we've already said that the photosynthesis involves absorbing light and fixing carbon, right? But in fact, those two parts are separate parts of photosynthesis. There's the light reaction, which is the absorption of light energy, and then there is the fixation of carbon. Those are different parts of photosynthesis. They are coupled. They are coupled together. We're gonna in just very, very general terms see how they are separate yet coupled. So the light reaction happens in these little stacks of these discs inside of the chloroplast, which is a larger plant cell unit. Inside of it are these discs that absorb light. It takes water and is going to reduce it and produce oxygen. The oxygen is the output, but those hydrogens that are ripped off of the water are used as reduction equivalents for these molecules called ADP and NADP+. So NADP+ is a coenzyme that we'll talk about in much more detail later when we get to the animal side of the equation. But it's essentially just an electron carrier. It's an electron carrier. So the NAD+ is positively charged. It gets protonated and is able to bring those electrons along with it. And as is ATP, ADP gets converted to ATP. ATP is an energy source. I just said that glucose is the fundamental currency of energy in the body, but maybe not fundamental is the right word. Glucose is like the $100 bill of a cell. ATP is the $5 bill. All right. So ATP is in units that are usable. You don't every day go to buy lunch with a $100 bill. You can break a $100 bill up into a bunch of $5 bills and spend those to buy your apples or buy your burrito or whatever you're buying. Okay, so the energy from the light reaction is used to get the reduction equivalent out of the NADP+ and to create energy in the form of ATP. So it takes ADP, adenosine diphosphate, adds another phosphorus onto it, an inorganic phosphate, to make it ATP. We'll see what those look like in a bit. Not right now. Don't worry about that. We'll get to them. Anyways, these two things drive the dark reaction, the reaction that's not dependent upon light. It's called the Calvin cycle. The Calvin cycle is what's responsible for taking carbon dioxide and making carbon-carbon bonds with it and carbon-hydrogen bonds out of CO2. So the input to the Calvin cycle is carbon dioxide, and the output is a sugar molecule. And then also, once that reaction has happened, it kicks out the NADP plus, which has given up its proton, and the ADP, which had released its energy in terms of the phosphodiester bond in ATP that had been hydrolyzed. We'll see what those look like in chemical terms in a little bit. All right. Are there any questions on this, on the overall structure of that? All right, here's C3 and C4. C3 plants are plants that grow mostly in this climate. At least they evolved to grow in this climate. Temperate, cool season plants. These are plants that do well in the spring and the fall, or plants at more northerly climes. Plants that have a decent amount of rainfall. They tend to make a higher percentage of protein than C4 plants. C3 plants, like peas, for example, or beans, spinach, kale, broccoli, these kinds of plants, they're pretty high in protein, higher in protein than corn, for sure. Yeah, Damon. Green. Many of them are green, but I mean, all of the plants we're going to be talking about here are green because they're photosynthesizing. Yeah. So there are different isotopes of carbon, and the next sequence of lectures, I'm putting my basic chemistry. There's going to be one lecture of basic chemistry for you guys where I talk about isotopes and all that. I probably should have put it earlier in the semester before this. But an isotope, we'll get to it, but an isotope is essentially an atom of a certain element, say carbon, for example. In this case, carbon. Carbon can have, carbon is carbon because it has 6 protons in its nucleus, but it can have a variable number of neutrons. It can have 6 neutrons, and that's carbon-12, or it can have 7 neutrons, and that's carbon-13. The 6 plus 6 protons, 6 neutrons, carbon-12, 6 protons, 7 neutrons, that's carbon-13. Those are different isotopes of carbon. Because they have slightly different masses, the kinetics of the reactions that they take place in, and particularly carbon fixation, carbon are a little bit different between between carbon-13 and carbon-14. Because the pathways are different in C3 and C4 plants, there's a more pronounced carbon isotope separation in C3 plants. So if you look at the carbon isotope ratios and fingerprints of C3 versus C4, they're going to be really different. And that deviation from natural abundance is more pronounced in C3 plants than C4 plants. Yeah?
So in C3 plants, you should have leaves, like multiple leaves on one branch. C4, you can have
C3 and C4, they're showing these idealized pictures, like a C4 plant there that looks like that's a corn thing, and they have an elm plant on the left. I've never eaten an elm leaf, but I have eaten kale, and kale leaves don't look like that, and broccoli doesn't look like that, and et cetera, et cetera. So C3, these are like incredibly enormous categories of plants that cover all. There's only one other type of plant out there called cam, and that's essentially cactuses. So anything that's not like a euphorbia or a cactus is going to fall into this. So making generalizations about leaf patterns is a little bit hard to do. Yeah. The generalizations that you can make are up here. C3 plants tend to be grown in northern climates. C4 plants tend to come from tropical places. There's plenty of C4 plants that have that kind of leaf distribution in the tropics. All right. So C4 plants are much more efficient at gathering carbon dioxide and using nitrogen from the atmosphere and the soil. Importantly, they take, pardon me, much less water to make the dry matter of the plant. And the heavier carbon-13 is less depleted in C4 plants. So the carbon isotope ratio in C4 plants is closer to natural abundance, the natural abundance that you would see in nature. All right, but before I flash forward, any questions about C3? The take-homes are not in the diagram. The take-homes are in those bullet points on the side. Temperate cool season, tropical warm season, carbon isotope separation more pronounced. In C3, carbon, more efficient carbon and nitrogen utilization in C4, uses less water, and less of the C13 is depleted. There's a higher ratio of C13 in the C4 plants. Yeah. It's like super loud, my friend. Dry matter means the plant we're going to find out in lab. I guess it's going to be next week that we take the apples that we're going to work up on Friday, and then we're going to figure out what the dry matter is the following week. But it's the part of the plant that has been completely dehydrated. If you desiccate plant matter or anything and take out all the water, the mass that's left is the dry matter. Oops. Oh yeah, no, you're just hanging out. Carbon cycle. So in the carbon cycle, there's a lot of different cyclings, and there's a kind of a hierarchy of these subcycles that you see. These different subcycles are going to run on different time scales, right? So you can have the range of time scales can go anywhere from much longer than 1 million years to very rapid, within just years, a few years, okay? So the slow carbon cycling is about 1,000 to 1 million years, and that's primarily like inorganic carbon, like carbonate, that's moving through rocks in much the same way as phosphorus might, all right? However, the fast carbon cycle is the cycling of carbon that involves a gaseous form, right? We said that phosphorus is different than carbon and nitrogen because it doesn't have any kind of significant gaseous form. There is phosphine, but that's not really important to our discussion. In general, phosphorus doesn't have a gaseous form. Carbon does. It's carbon dioxide. We all know about it. Everybody and their brother knows about climate change, whether they believe it in or not. There certainly is carbon dioxide in the atmosphere. That carbon dioxide be taken up by plants. The plant matter can get consumed by an animal. The animal can die or leave feces. And that can go into the soil and be liberated as methane, perhaps. Or the animal consumes that plant matter, combusts it in cellular respiration, and breathes the carbon out again as carbon dioxide. So, for example, you could, in your dorm room, grow some sprouts. Anyone ever sprouted before? You could grow some sprouts in your dorm room. Those sprouts fixed the carbon. You could eat the sprouts and then go jog. And as you jog, you're burning the energy that you absorb from those sprouts, the carbohydrates, and exhaling that carbon as carbon dioxide. So there's it would take you a few days to grow sprouts. That's a carbon cycle of a few days, all right? So carbon can cycle quite quickly. through the ecosystem. All right. All right, here is breaking the carbon cycle down more abstractly, not on a landscape scale, but in more of a chemical scale. So on the left, you see organic carbon. This is carbon that you would see this is carbon that's being utilized through heterotrophs, right? So, for example, the sprouts would be organic carbon. Sugars, any kind of organic carbon source, plants, animals, those are sources of organic carbon that heterotrophs, which are us, anything that's not a plant that's photosynthesizing, us, are going to eat that and can turn over that organic carbon. Then there's also respiration that happens, where we take organic carbon, the sugars that you consume, and then respire with it, producing inorganic carbon. All right, so that's the respiration arrow that's going from organic carbon to inorganic carbon. Plants do the other side. They come back and do the oxygenic photosynthesis. So I've talked about everything on this. There's this dotted line that goes across that. That's all the oxic processes that involve oxygen. All right? So you eat sugars, you ingest them, they go to your cells combust them through cellular respiration, and then you breathe out the carbon dioxide. Plants take it up and fix it again. That's this back and forth between inorganic and organic carbon on the top half of this diagram. Those are oxygenic or oxic processes. There's also anoxic processes that involve carbon. For example, fermentation, the fermentation we're doing. So you've got your sauerkraut, you've got this weight on top of it, keeping all of the sugars in your brew underneath the water, underneath the acidic, the slowly acidic, slowly, rapidly, I don't know how quickly, but acidifying solution, keeping it away from the oxygen. It's that organic carbon that can be used to make inorganic carbon as well. This is the carbon dioxide, for example, in the SCOBY, in your kombucha. Your kombucha can get a little bit bubbly. And that's what's we're gonna do in the fermented food labs next week. We're gonna bottle our kombucha and put it under pressure, and it's gonna build up carbon dioxide through this anoxic process. We're gonna fill the bottle, cap it, keeping oxygen out. It's gonna produce carbon dioxide, and it's gonna be dissolved in the water through this anoxic process. We're going to take organic carbon, the sugars in the kombucha, and we're going to produce inorganic carbon or carbon dioxide or carbonate, right? Carbonic acid. There is, so some of the other arrows here, the ones that involve methane are not as important to us in this class until we get to the gut. So this week, you are listening to the Brains On podcast, and the other one is it a Radio Lab, a This American Life? Which is it? Radio Lab. It's Radio Lab, yeah. It's all about farts. So that is all of the parts of that carbon cycle in the middle that involve methanogenesis, all right? So taking organic carbon, the sugars and the different starches in the food you eat, maybe the soluble or insoluble fiber and methanogens in your gut that are not exposed to oxygen, bacteria that are not exposed to oxygen, produce methane gas in there. And that's what your farts are. That's what your farts are. So it's that part of the carbon cycle that we're talking about in the podcasts this week. All right?
Isn't methane flammable?
Yes, it is flammable. I probably wise of me not to put a video up here, but sometime when you're bored on YouTube, I'm sure you can find some high schoolers passing the time by lighting their farts. I don't know if you've ever heard that, but people, you can light your farts on fire. I don't advise it, but I'm sure some of you have heard of that before. No? None of you? Yeah, okay. Of course, snails and puppy dog tail boys all know about lighting their farts. I don't know. Yeah.
How does it make it's like
Okay. Yeah. So the different types of foods that you eat are going to affect the flora in your gut. Certain bacteria like certain foods more than others, and so when you feed, so for example, my one son loves sausage. He like one of his battle cries is eternal sausage. He'll say that at dinner when I have sausage. Like he wants it, right? And if it weren't for him, the rest of the family would probably be vegetarian. My wife and I were. My daughter is not really into the idea of eating animals. But my son wants sausage. And so today is my son's birthday, right? He's turning 8, and after I get out of here, I got to go to the store and get some hamburger because he wants burgers and get some sausage because he wants sausage for dinner. But I also have to get some veggie burgers and veggie sausages to keep my daughter happy and my wife happy because they don't want hamburgers and sausages. So I'm going to have different foods that different organisms, not that my daughter and son are different organisms, but they are different organisms, same species. They're going to respond differently to different kinds of foods. All right. Some people get gassy when they eat certain beans. Not everybody. There are enzymes that help proteases that some people have that can break down the proteins in a bean, and other people don't have it as much, so much of that. And so whatever it is, the specifics of those reactions that are happening in the gut, sometimes beans are producing fuel that methanogens particularly like, so it can produce a lot of gas. Okay? Some people get gassy with cabbage, which I'm sure is exciting for those people that were making sauerkraut. Although sauerkraut is a typically less methanogenic form of cabbage than raw cabbage, so maybe you won't be as gassy. Some people get gassy with meat. Some people get but bean is a common one. Does that answer your question? Any other questions about farts? This is all I'm really going to say about farts today, but that is the podcast. All right. All right. So, the carbon cycle. Still on the carbon cycle. So I said there's this hierarchy of subcycles. This is a much more sophisticated, landscape view of carbon. And this is the global carbon cycle. You can see on there all these numbers. Those represent fluxes of carbon. So the tubes are fluxes of carbon. And the number in the boxes are carbon reservoirs. And those numbers are measured in gigatons of carbon. So you want to get to a steady state. People often confuse the terms equilibrium and steady state. Equilibrium is a dynamic system that is not getting any kind of energetic input. All right. A steady state is a dynamic system that is getting an energetic input, and but where the dynamics are not changing. All right, that's a steady state. Equilibrium is a steady state, or it is a situation where the dynamics are not changing, but there's no energy input. All right, so it's, for example, a body or a cell at equilibrium is dead. A living body is at steady state. You guys get the distinction? So the planet is actually alive, or at least the things on it are alive. The planet itself is debatably alive, but it's at steady state. So to maintain a steady state for any of these reservoirs, to maintain a steady state in those reservoirs, the inputs into a reservoir need to equal the output of a reservoir. Inflows and outflows must be coupled to maintain a steady state. The problem currently is that the inflow and outflow of carbon in the global carbon cycle has been decoupled, and we are not at a steady state any longer. They've been decoupled. That's basically by that blue arrow there, that's the steady state has been disrupted by the burning of fossil fuels. We've been taking a huge amount of carbon out of the geologic reservoir, these sedimentary rocks, which are fossil fuels. We burn it, we chuck the carbon dioxide up in the atmosphere, and that's throwing off the steady state with the carbon dioxide that's in the atmosphere. But that reservoir is connected to all these other reservoirs. So it's changing the size of the other reservoirs in the carbon cycle. Methane reservoir is going up. There's more methane, which is like 40 times or 100 times or 400 times, I forget the number, but it's radically more potent as a greenhouse gas than carbon dioxide. Methane is going up in the atmosphere. But it's also affecting primary producers. The primary producers and the reservoir of carbon that's there. We're going to see how in just a bit. That's the last point I want to make in the class today. And that, in turn, is attached to consumers, us, the carbon in us, and how that's throwing everything off. So here on the right is just a diagram of the carbon CO2 levels of the last 10,000 years. For those of you who, there probably is nobody in this class, but for those of you who don't think carbon dioxide is going up, it is. And there's the trace. So, here's a different way of looking at, yeah, Nick. Super loud.
Why are cows responsible for producing so much methane?
Oh, because there's a lot of methanogens in their ruminants, and they eat grass. And so the particular chemical pathways that allow... So I talked about earlier cellulose, how hard it is to hydrolyze the glycosidic bond in cellulose, right? The beta-1,4 linkage. Wood does rot, right? Wood does break down. It's just very slow. The kinetics of it are very slow. So cows are ruminants. They eat that cellulose, the grass or whatever, and it has to sit. To ruminate onto something is to like sit there and mull it over or think about it, right? But ruminants have like 4 stomachs. The grass goes in there. It goes from one stomach to the other and just sits there forever and ever. The particulars of digestion in their digestive tracts are skewed towards methanogens. And so there's a lot of methanogenesis that happens from the organic carbon in their GI tracts. And then they produce a ton of methane. Cows fart a lot. They're also big, so they're eating more. Anyone else? Okay. Let's see if I can set us up for next time. I had wanted to finish the carbon cycle today. I'm always about 5 slides behind. So this is kind of just another way of looking at that diagram on the left in the last slide, that global carbon cycle. It's looking at different carbon sinks and reservoirs in the atmosphere. So forest land takes up a small amount, cropland takes up a small amount. There are other ways of carbon fixation, that carbon fixation happens on the planet. But fossil fuels are way offsetting that by chucking up huge amounts of carbon dioxide into the atmosphere. So the gray is much larger than the green there. And as a result, here's some fine-scale data of carbon dioxide that's being measured at Mauna Loa, which is a mountain out in Hawaii, a very remote location, to isolate it from industrial effects and to look at the global carbon rise. So carbon dioxide is going up, folks, and it has a real impact. So this is the last concept, and then I'm going to hit the consequence of this next time. I got about a minute. As atmospheric carbon dioxide goes up, plants kind of like it because there's more carbon dioxide for them to absorb. So I said a steady state is a system where inflows match outflows. If you think of a plant and its photosynthesis as a steady state, if you're pushing more carbon dioxide into it, right, just because there's more available, the plant's going to go to steady state. You're going to be getting more carbon fixation, right? Sounds like a good thing, hey? Sounds like a good thing. Well, we'll see the consequences of it next time. But we look at this here, carbon dioxide concentration. As the carbon dioxide concentration goes up, photosynthesis also goes up. So that curve there is showing the difference between C4 and C3 plants and how much they photosynthesize as a function of partial pressure of carbon dioxide. So for next time, we're going to pick up with this question. I want you to think about this slide. The slide is on Moodle, or you can look at it in the lecture, but this basic relationship. And we're going to start by talking about aside from the effect of carbon dioxide on climate change, is it a good thing? Tell me the pros or cons. I don't know. You tell me. Is it a good thing or a bad thing that plants photosynthesize more rapidly with increased carbon dioxide, partial pressure? Think about it. That's where we're going to start on Friday. Questions? Is everyone happy, in a good mood? No? Yes? A little hot? Be in a good mood. You guys are young, you're smart, you get to take a class with me. What more could you want? Huh? Nothing? Good, good.